Hey again, Paul DiLucci here with another video presentation for our Unit 3 discussion. Today I'm going to be discussing a paper written by an Italian geneticist named Giorgio Bernardi and his theories on non-codon regions embedded into our DNA. So let's just jump right into it, and I'm going to discuss a little bit about the non-codon DNA mystery. Now this theory or this mystery is a profound and complex dilemma which has eluded scientists since the 1960s. Now, through the Human Genome Project and other various reaches of genetic research, it's now known that the human genome is composed of 21 or over 21,000 functional protein codon genes. However, these 21,000 genes only roughly make up about 2% of the entire human genome. Since the 1960s, there's been several various and differing models of how and why non-codon regions exist. The ENCODE project, which is a project tracking some of these non-codon genes, has projected that up to 80% of all those non-codon DNA is at least reactive in biochemical expression to some degree. But because of some of what they term as loose definitions of the reactivity which they're tracking, there really isn't an established or solid evidence that it's actually as high as 80%. Now, Bernardi uncovered a compositional non-uniformity of DNA fragments, noting or calling them isochore regions, which present with either a CG-rich or a CG-poor composition. These groupings are associated with various states of DNA or gene expression, where CG-rich isochores are associated with early replicating chromatin, open chromatin, and the CG-poor isochore regions are associated with late replicating and closed chromatin domains. The second component of Bernardi's theory involves not just the composition of the isochores, but how the isochores actually conserve the structure of the interphase chromatin. When mapped out or visualized in chromatin, Bernardi presented CG-poor regions, which loop around CG-rich regions, kind of looped in a flower-like, with the CG-rich being the inner portion. Now, during metaphase, though, they're all stretched out in one thin line, which with varying spaces of CG-rich with lots of CG-poor regions in between. There's a lot to be concerned about these mostly non-codon regions found within our genomic DNA. Now, it's very possible that evolution has not yet mutated to excise these regions, some of these regions being introns or exons. But there's also other sources, such as transposons, which are critical, which may be critical in the expression of, or critical in gene expression, if not at the very least influential. Now, after some additional research, I found that there are several articles which do not support Bernardi's theories in the isochore theory. But I think as we've learned anything through genetics and through scientific theory, that patterns sometimes mean more than what can be thought of, or that some of these CG-rich isochores actually do hold value in genetic research and theory, but just have not yet been able to be applied practically. So that is our discussion for today, and thanks for tuning in, and see you next time.